Good morning, everyone, and welcome. I am pleased to be here today to present on the topic of Dr. S.K. Singh's USP guidelines for quality control. Today's session will cover the key principles and best practices outlined in these guidelines. We will explore what makes quality control effective and how these standards can be implemented in our work. I look forward to sharing insights and engaging with you throughout this presentation. Let's get started. Let's talk about introduction to quality control. Quality control, or QC, is a crucial part of our process because it ensures that medicines are safe, effective, and consistent. Every batch of medicine we produce must meet specific standards that we've set beforehand, and these standards are non-negotiable. Think of QC as the final checkpoint before the products reach patients. It's where we double-check that everything is up to par. For example, if a batch of tablets fails a dissolution test, meaning it doesn't release the right amount of active ingredient, we reject that batch. This step is vital because it helps us avoid delivering medicines that might not work properly or could potentially be unsafe. Overall, QC acts as a safeguard, making sure that only quality medicines make it to the people who need them most. So what exactly is USP? Well, USP stands for the United States Pharmacopoeia, which is a global standard-setting organization. Essentially, it develops and maintains quality standards for medicines, supplements, and other healthcare products worldwide. They publish what's called the USP, NF, or United States Pharmacopoeia, National Formulary, which is a legally recognized compendium. These standards are not just guidelines. They have legal standing, meaning products must meet these criteria to be recognized as safe and effective. The standards set by USP apply across a variety of products, including pharmaceuticals, biologics, and dietary supplements. For example, if we look at paracetamol tablets, the USP monograph for paracetamol defines the specific quality standards these tablets must meet. This includes things like purity, potency, and acceptable appearance, basically ensuring that consumers get a consistent and reliable product. Overall, USP plays a crucial role in ensuring product quality and safety, helping to protect public health and support the integrity of the healthcare system worldwide. Let's talk about the importance of USP in quality control. First, it provides uniform testing methods worldwide, which is crucial for consistency across different regions. When we have standardized methods, we can be confident that tests are comparable regardless of where they are performed. This standardization helps ensure key quality attributes such as identity, strength, purity, and performance of the drug. For example, two companies manufacturing the same medication need to meet the same USP limits, ensuring that patients receive a product that is both safe and effective. Overall, the USP acts as a global benchmark, maintaining high-quality standards and supporting the integrity of pharmaceutical products across the industry. Let's take a look at the structure of USP standards. First, we have monographs, which detail the specific requirements for individual products. For example, an aspirin monograph will specify the acceptable assay range, such as 95 to 105%, to ensure the product's quality and potency. Then, there are the general chapters, which provide standard test procedures that apply across various products. These chapters help ensure consistency and reliability in testing methods. Finally, we have reference standards, which are verified materials used during testing to ensure accuracy and consistency. These reference standards serve as benchmarks, helping laboratories produce valid and comparable results. Overall, this structure, monographs, general chapters, and reference standards, forms the backbone of USP's approach to maintaining high-quality pharmaceutical standards. Let's talk about identity testing, which is a critical step in ensuring we have the right drug substance. This process helps us confirm that the product is exactly what it's supposed to be, preventing mix-ups and the risk of counterfeit products entering the supply chain. To do this, we use specific techniques. One of the most common is IR spectroscopy, or infrared spectroscopy, which allows us to analyze the sample's molecular vibrations. Another key method is HPLC, or high-performance liquid chromatography, where we compare the retention time of our sample to a reference standard. If they match, it's a strong indication that the substances are identical. For example, in IR spectroscopy, we look at the spectrum of our sample. If it matches the USP reference spectrum, we can confidently say that our sample has the correct identity. This kind of testing is essential for maintaining quality and safety throughout the drug manufacturing process. Now, let's talk about the assay, or drug strength, which is really about determining the amount of active ingredient in a medication. This step is crucial because it helps us make sure that the dosage is correct, ensuring the medication will have the intended therapeutic effect. There are a couple of common methods used for this purpose. One is titration, a classic chemical technique that helps us find the precise concentration of the active ingredient. The other is high-performance liquid chromatography, or HPLC, which is a more advanced method that can separate and quantify components with high accuracy. For example, if we have a 500 mg tablet, regulatory standards typically require that it contains somewhere between 95 and 105% of the labeled drug amount. That way, we can be confident that the medication is neither underdosed nor overdosed, maintaining safety and efficacy. Overall, ensuring the correct drug strength is a key part of quality control in pharmaceuticals, tying into the broader theme of safety and effectiveness in medication production. Now, let's talk about impurity testing. This is a crucial part of ensuring the safety and quality of pharmaceutical products. Impurity testing helps us detect any harmful or unwanted substances that might be present in the drug. These impurities can come from various sources, such as manufacturing processes or raw materials. Specifically, we look for related substances, residual solvents, and heavy metals. Related substances are chemical impurities that are closely related to the active ingredient, and they need to be carefully monitored. Residual solvents are leftover chemicals from the manufacturing process, and it's important to keep them within safe limits. For example, residual methanol must be below the limits set by the USP to ensure safety for patients. Heavy metals are another concern, as they can be toxic even in small amounts. Overall, impurity testing helps us confirm that the product is safe, effective, and compliant with regulatory standards. It's a key step in maintaining the integrity of the pharmaceutical supply chain and protecting patient health. Now, let's talk about dissolution testing. This is a crucial part of pharmaceutical development because it measures how quickly a drug dissolves in the body. You see, the rate at which a drug dissolves can significantly impact its effectiveness. If a drug dissolves too slowly, it might not reach the necessary concentration in the bloodstream to have its intended effect. Conversely, if it dissolves too quickly, it could cause issues like irritation or rapid absorption that might not be desirable. This testing is especially important for ensuring bioavailability, which is essentially how much of the drug actually becomes available in the body to produce its effect. For example, with immediate-release tablets, we typically expect them to dissolve within about 30 minutes. That quick dissolution ensures that the drug is absorbed efficiently. But what happens if the tablet doesn't dissolve properly? Well, failure in dissolution can lead to reduced drug absorption, meaning less of the drug gets into the bloodstream, and, ultimately, less of the therapeutic effect. So, maintaining proper dissolution rates is vital for both safety and efficacy. Overall, dissolution testing helps us confirm that our drug formulations will perform as intended once they are taken by patients. Let's talk about the uniformity of dosage units. This is really important because it ensures that each dosage unit, like a tablet or capsule, contains a consistent amount of the drug. Consistency here is key for safety and efficacy. To achieve this, we use specific methods, mainly weight variation and content uniformity. Weight variation involves checking if the individual units are similar in weight, which generally correlates with the amount of drug inside. Content uniformity, on the other hand, directly measures the amount of active drug in each unit to make sure they match up closely. For example, when we look at capsules from the same batch, they shouldn't vary significantly in their drug content. If one capsule has much more or less drug than another, it could lead to ineffective treatment or potential side effects. So, maintaining uniformity helps us guarantee that every patient gets the right dose every time, which is the foundation of quality control in pharmaceutical manufacturing. Now, let's talk about microbial quality control. This is a critical part of ensuring the safety and effectiveness of pharmaceutical products. Its main goal is to prevent contamination by harmful microorganisms that could compromise the product or pose health risks. To achieve this, we perform specific tests. One is the total microbial count, which helps us understand the overall level of microorganisms present. The other is sterility testing, which is particularly important for injectable drugs. For example, injectable drugs must pass sterility tests, meaning there should be no microbial growth in the sample. If any microbes are detected, the product cannot be used, as it could cause infections or other complications. Overall, microbial quality control is essential because it safeguards patients and maintains the integrity of our pharmaceutical products. It's a key part of the quality assurance process that underpins trust in our medicines. Let's look at an example. A high-performance liquid chromatography, or HPLC, method must consistently produce the same results when we repeat the tests. This consistency is crucial because it confirms the reliability of our testing methods. When we're working with USP reference standards, we're dealing with highly purified substances that serve as benchmarks for accuracy. These standards are essential for several uses, such as calibrating our instruments to ensure they're working correctly and validating our methods to confirm they produce accurate and reliable results. For example, in chromatography, we compare the sample peak with the USP standard to verify the sample's identity and quality. Now, when we talk about analytical method validation, specifically according to USP 1225, there are key parameters to consider. These include accuracy, which tells us how close our measurements are to the true value; precision, which assesses the reproducibility of our results; specificity, ensuring our method accurately measures the analyte without interference; linearity, confirming that our method provides proportional responses across a range of concentrations. All these components work together to ensure our testing methods are robust, reliable, and compliant with regulatory standards, ultimately supporting the quality and safety of pharmaceutical products. Let's talk about documentation and compliance. It's essential to maintain accurate and complete records throughout every stage of our process. This ensures that we have a clear and reliable trail of what was done, when, and by whom. Following good laboratory practices, or GLP, is also critical. It helps us uphold quality and consistency in our work. By adhering to these standards, we make sure that everything is traceable and audit-ready at any time. For example, batch records are vital because they help us track the entire history of a product. If there's ever a need for a product recall, these records allow us to quickly identify the root cause and address any issues efficiently. Overall, solid documentation and compliance are the backbone of a trustworthy and transparent operation, giving us confidence in our product quality and regulatory readiness. Let's wrap up with some key takeaways. First, following USP guidelines is crucial because it ensures that our quality standards are consistent everywhere in the world. This consistency is vital not only for meeting regulatory requirements, but also for ensuring patient safety. When we adhere to these guidelines, we're helping to guarantee that every product is safe, effective, and reliable. And remember, quality control isn't just about testing. It's about actively assuring quality throughout the process. It's a comprehensive approach that supports continuous improvement and trust. Finally, I want to leave you with this thought. Every tested batch truly represents a patient's trust. When we do our part diligently, we're not just meeting standards, we're safeguarding lives and maintaining confidence in our products. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. Remember, our goal is to ensure you feel confident and well-informed moving forward. Thank you so much for your attention and participation today. We truly appreciate your time and engagement.